Good morning. So we're going to start up with part 2 of your protein synthesis lecture. Yesterday we talked about the first step, which was transcription, which was the copying of your DNA onto the messenger RNA. That's where we last left off. And we just did some extra processing with the eukaryotic cells by cutting out the introns and splicing back the exons. So now we have our messenger RNA that has been sent out into the cytoplasm. So now we'll pick up with the second half of protein synthesis, which is translation. This occurs in the cytoplasm of your cell. We talked yesterday a little bit about codons. Those are those sequences of 3 bases on your messenger RNA. There's also going to be anticodons. I think I mentioned that yesterday. So with that, your transfer RNA is going to have these anticodons. They are going to match up with your codons that are on your messenger RNA. So if your messenger RNA says AUG, your transfer RNA would be UAC. That's what it's going to read. So the main job of translation is now to translate your genetic code, specifically your messenger RNA, into the language of proteins by building chains of amino acids. So I already said this, but your transfer RNA anticodons complement those messenger RNA codons. So the main thing that I also want you to remember with transfer RNA, they are going to carry amino acids. So their anticodon sequence is very specific to the amino acid they pick. For example, the one that I used, which was UAC on the transfer RNA, that will always carry the amino acid methionine, always. Alrighty, so apparently I have this in the middle. I'll move off to the side for just a moment. Process of translation. Your first step here really is that your messenger RNA goes to find your ribosome. Kind of a refresher, your ribosome has a large subunit up here and a smaller subunit down here. These are sites. They go by letters. I'm not going to have you know which letter each one goes to. Actually, there are 3 different sites, and each one reads one specific codon. I don't know if I said bases. If I said bases earlier, I apologize. Each site here will read a specific codon. So, and the messenger RNA is going to feed right through here where the small subunit and the large subunit meet. So, so your messenger RNA is going to come through here. Your transfer RNA is going to kind of meet up with it as they are getting right here in this center site. There are about 30 naturally occurring amino acids. Like I said, they're very specific to their anticodon. And so as your body breaks down your food, specifically breaks down the protein that you eat, it's going to break apart the protein into those individual amino acids. Those amino acids then get brought into your cytoplasm. Your transfer RNA molecules then pick up their specific amino acid from there. So that's where they're getting those amino acids from. And then they will bring them over to the cytoplasm to attach here to the messenger RNA to start building that protein. This is kind of what your transfer RNA looks like. It's kind of a weird looking thing, but sometimes people will call it a hairpin shaped molecule. I don't know if it looks like a hairpin, but so this is your RNA. Kind of folds itself here. These, if you notice, are complementing these bases right here and these bases right here. That is specific. That's so they can form this particular shape. these down here, these three right here, these are the anticodon. And they're going to match up with this codon down here on this messenger RNA. So you can see that the codon reads GCC, so the anticodon is CGG. This particular one is carrying this amino acid. It seems to be a generic amino acid. So if you recall back to the protein, the macromolecules, when we talked about the proteins, they had that basic structure with the carboxyl group and then the amine group and a carbon and this R group. So the R group is what makes your amino acid different, because everything else will be the same. Not that you'll need to know this, it's just more of an FYI. The amino acid is held onto your transfer RNA molecule by an ester bond here. So once these connect, these are able to connect to the other amino acid that's right before it via a peptide bond, and then that ester bond is broken, and then the transfer RNA molecule will leave. I think I've got more pictures though back to here in just a moment. So, alrighty, so the messenger RNA has now gone to the ribosome. The transfer RNA then is going to come over to the ribosome and match up with the messenger RNA. So those anticodons are matched up with the codons. The ribosomes can only read one codon at a time. However, like I said, there are three quote-unquote sites. It's only going to read that center one, but there's going to be some prep work done in the first site. It's going to read it in the second site. In the third site, it's going to be forming that peptide bond. So even though there's technically three codons in the ribosome, it's only reading one of them at a time. We call this the reading frame. Every single protein starts with this sequence right here, this AUG, which I said codes for methionine. So the AUG is what's on the messenger RNA. What's on the transfer RNA then is going to be UAC. That particular transfer RNA will always carry the amino acid methionine. And it's going to keep going ahead and processing and pushing that messenger RNA through the ribosome will move along. Actually, it doesn't feed through the ribosome, moves along the messenger RNA, not the other way around. The messenger RNA doesn't get pushed through the ribosome. But the ribosome will continue down that messenger RNA strand and keep reading it until it gets to a stop codon. There are, I believe, 3 or 4 specific sequences that signal the end. So they don't code for an amino acid. I know one of them is UAG. So or UGA, sorry. When the ribosome comes across the UGA, there is no amino acid, but it just knows to stop. And so the ribosome then stops translating, detaches itself from the messenger RNA, and the protein is released from the ribosome to go out and do whatever it is it needs to do, whether it's to create pigment for your eye color or whatever. So like I said, there aren't any transfer RNA codons. So it just knows to stop. I don't know if that makes sense. I hope it does. Okay, so this is kind of a better picture here, kind of like what I was talking about. So here, they're not showing the entire ribosome here. They're just showing the bottom part of the ribosome, but it's feeding through sorry, that was our dog. We're feeding through the messenger RNA strand here, and it's lined up with that start codon, and this is a very interesting representation of a transfer RNA, but You can see that it's holding on to the methionine here. All of your amino acids, by the way, are abbreviated with three letters. And methionine's abbreviation is MET. And then what will happen is it will start moving down through the different sites. So this is the binding site. So then what will happen is they'll start reading it that way, and it will read these next three bases, or that next codon. This is kind of what I was alluding to. The three different places, they're the different sites, the E site, the P site, and the A site. So like I said, you probably won't need to know about those sites until you get to college, deep into college. So I'm not going to go too much into depth on that. Just understand that there are three different places, but this is the only part that's actually being read. The P site right here in the middle is the only codon that's actually being read by the ribosome. So this kind of shows you too. The transfer RNA is reading. It's got the methionine, so it's got it right here, and then the transfer RNA is coming in here to this A site, and so what they're going to do is then this one sits down, and then and in this case, actually, I think they've got it backwards. Shouldn't be 3 to 5. It should be 5 to 3. And then it's going to, so then your ribosome is going to read this next three set of bases right here, the GCC, which codes for alanine. And then the methionine and the alanine are going to get a peptide bond formed. And then this RNA strand is going to detach itself, so it's not empty, and it's going to leave. And then a new transfer RNA is going to come into this A site and attach. This one looks like it's tryptophan. So then that's going to attach here to the alanine. And you can see that you're starting to build your protein because it's just a sequence or a chain of amino acids. And then you're just going to keep going and adding more and more until you get to a stop codon, in which case you'll be done. And then these chains of amino acids will detach from the ribosome. So they'll once they detach, they'll be able to form that alpha helix or beta pleated sheet. But right now it's technically just the primary structure, which is the sequence of the amino acids. So, okay, last thing, just like with transcription, you can have multiple ribosomes working on a single strand of messenger RNA. Your genetic code is degenerate, meaning there's a lot of amino acids that will have more than one codon. On Monday, I will kind of explain to you how to read the table that goes with it, and you'll see that there are several, usually four different codons are going to code for the same exact amino acid. And usually the only thing that's different is they're going to have the first and the second nucleotides, or sorry, the first and second bases will be the same. The only thing that will be different is that third base in the codon. So that's why there's 4, since there's 4 different bases. And again, this probably doesn't make a lot of sense. It should make sense when I show you the table on Monday. The reason why we have this in place is so that if for some reason your ribosome reads it as CCA instead of CCG, those two codons are going to code for the exact same amino acid. So the hope is that you will be able to prevent some mutations, because while it's technically a mistake, it's still going to code for the same protein. So you're going to be okay. We'll talk probably next week about how mutations can affect a protein. So for the most part, the genetic code really is universal among organisms. Every organism will do transcription and translation this way. The only exception is the mitochondrial and chloroplast DNA in mammals. It's a little bit different, but we won't go into that. Just understand that every single other organism transcribes and translates this way, except if the DNA is in the mitochondria and the chloroplast. So that kind of wraps up translation and transcription. This is basically how a protein gets built. Like I said, next week we'll get into how you're building the protein and we'll practice doing transcription and doing translation. So it's a pretty simple process once you get it under your belt, though. So I'm sure you'll be good. But again, like always, if you have any questions, please let me know.